Dear students, welcome to the fifth session of Histology, Animal Behavior and Applied Zoology paper prescribed by the Mangalore University for 4th semester BSc. In this session, we shall consider the histology of mammalian ovary. As we all know that the mammalian ovaries are the female reproductive organs. A pair of ovaries will be generally present in all mammals, which will possess many follicles at different stages of their growth, and a mature ovum will be periodically expelled out, making it available for fertilization. Histologically speaking, the ovaries of mammals, in particular the human ovaries which we are referring here, will have two superficially demarcated structures called ovarian cortex and ovarian medulla. Though these are not clearly demarcated histologically, but under microscope we can easily differentiate the cortex from medulla. The cortex is a structure which has the growing follicles, the attriting follicles, as well as carpus luteum and carpus albicans. The medulla of the ovary consists of connective tissue and blood vessels. So histologically, the ovaries are covered by a simple cuboidal epithelium called the germinal epithelium. This is the outermost part. Okay, this region is called the epithelial covering or germinal epithelium. Deep to the germinal epithelium, the ovaries possess a capsule called the tunica albuginea, the covering of the ovaries, that are composed of a dense, irregular, collagenous connective tissue. Each ovary is subdivided into a cortex, this part is the cortex, and a medulla, which are not sharply delineated. The ovarian cortex consists of ovarian follicles in various stages of their development and a connective tissue stroma containing cells that respond in unique ways to the hormonal stimuli. So as you can see here, the cortex of the ovary is fully composed of the follicles at different stages of their growth and development. So the follicles, which are in the very initial stages of their development, are called primordial follicles. So these small follicles which you can see here are called primordial follicles. These primordial follicles are composed of a primary oocyte. So there will be an oocyte in it, enveloped by a single layer of flat follicular cells. So you can see here the primordial follicles are simply the primordial oocyte, a primary oocyte covered by a single layer of flat follicular cells. So this is the oocyte surrounded by the single basement layer or basement lamina or granulosa cells. The next stage in the development of the oocyte is the primary oocyte. So these primary oocytes display a prominent eccentric vesicular appearing nucleus possessing a single nucleolus. The primary oocytes have many Golgi complexes, mitochondria, and profiles of rough endoplasmic reticulum and well-developed annulate lamellae. They become arrested in the prophase of the meiosis first by paracrine factors produced by the follicular cells during the fetal life and remain in this stage until ovulation, generally perhaps for many years. The secondary follicles are the next stage in the development of the ovarian follicles. These follicles are also called as antral follicles because a hollow space called antrum filled with a fluid folliculi gradually starts to develop in these secondary follicles. So secondary follicles are established when fluid, which is called folliculi, an exudate of plasma containing various hormones such as activin, estradiol, follistatin, inhibin, and progesterone, begins to accumulate in the spaces between granulosa cells. The fluid-filled spaces will begin to coalesce and eventually form a single large cavity called antrum. The secondary follicles are dependent on FSH, that is follicle stimulating hormone, for their growth and development. Narrow processes extend from the granulosa cells into the zona pellucida. Granulosa cells contact each other via gap junctions and also form gap junctions with the cell membrane of the primary oocyte. The fully mature ovarian follicle in the mammals is called Graafian follicle. So this is a structure of Graafian follicle. The dominant Graafian follicle is one follicle among the secondary follicles that will ovulate. It is FSH independent and manufactures the hormone inhibin that shuts off the FSH released by the basophils of the anterior pituitary, causing atresia of other developing follicles, secondary and non-dominant Graafian follicles. So this is very important because at a given time only one ovum can be expelled. So in order to suppress the multi-ovulation, the Graafian follicles, which are mature, will suppress the secretion of FSH so that the other cells which are growing will fall short of FSH and hence they become apoptotic. That means a programmed cell death will be activated in them, causing the death or degeneration of other follicles. This is just to reduce the competition among the growing follicles. The fully mature Graafian follicle measures approximately 2.5 centimeters in diameter and is evident as a large bulge on the surface of the ovary. So externally, if you look at the ovary, there will be a bulge which is clearly visible like edema, and that particular part will have the fully mature Graafian follicle. The Graafian follicle is externally covered by a layer of cells, keratinized cells called theca externa, which inside will have a fully grown mature oocyte, and this oocyte is covered by a heap of granulosa cells called cumulus oophorus. And as a result of much of all the antral fluid filled spaces, they form a large fluid filled space inside the membrana granulosa cells called antrum, filled with the liquor folliculi. And the layer immediately outside the mature oocyte is called zona pellucida. Zona pellucida. The primary oocyte is positioned off-center on a small mound of granulosa cells called cumulus oophorus, which we have already discussed, that projects into the liquor folliculi containing antrum of the follicle. Granulosa cells surround the zona pellucida. Those contacting the zona pellucida are known as corona radiata. Other granulosa cells line the antrum, forming membrana granulosa. So those granulosa cells which comes in contact with the zona pellucida are known as corona radiata cells. The theca interna cells, these are the cells of theca interna. You can see some brown colored cells here. They manufacture androgens which are transferred to granulosa cells where they are converted into estrogens. So the theca interna as well as the granulosa cells are very important for the process of production and secretion of estrogens. The theca externa is mostly collagenous, the outermost layer. It contains a few smooth muscle cells, many blood vessels which provide nourishment to the theca interna. The function of theca externa is to provide protection as well as to nourish the theca interna cells. Once the ovum is expelled out of the Graafian follicle, this generally happens at the 14th day of the menstrual cycle due to a surge in the luteinizing hormone, if at all the individual is carrying a perfect 28-day menstrual cycle. Once the ovum is expelled out of the Graafian follicle, the granulosa cells, theca externa cells, and all other cells of the Graafian follicle which remain are now termed as corpus luteum. The corpus luteum is formed from the corpus haemorrhagicus. Corpus haemorrhagicus is some structure which is formed after the expulsion of the ovum from the Graafian follicle. This is called corpus haemorrhageus because of the blood clot that is significantly observed in this region after the expulsion of the ovum. This corpus luteum is composed of granulosa lutein cells, which are modified granulosa cells, and theca lutein cells, which are modified theca interna cells. So that means the granulosa cells and theca interna cells of the Graafian follicle gets modified into lutein cells of granulosa or granulosa lutein cells and theca lutein cells. The formation of this richly vascularized temporary endocrine gland is dependent on luteinizing hormone secreted by the anterior pituitary or adenohypophysis. The function of corpus luteum is to secrete a hormone called progesterone, which is also considered as the pregnancy hormone. So once the ovum is expelled out, it will be available for fertilization for around 2 to 3 days, but not more than 48 hours in specific. So if there is fertilization, then corpus luteum goes on producing the hormone progesterone, which maintains the pregnancy until the childbirth. If there is no fertilization, then on 28th day, generally the corpus luteum stops secreting progesterone, as a result of which the endometrium of the uterus collapses, leading to something called menstruation. So once this happens, if there is no fertilization, no pregnancy, then the corpus luteum generally degenerates, forming a structure called corpus albicans, which could be seen in this figure as a degenerating corpus luteum. That's all about the histology of mammalian ovary. For further information, you can refer these textbooks, which are readily available in most of the libraries of our colleges. Thank you.